Proteins are these complex and critical little molecular machines that allow living organisms to, well, do everything that living organisms need to do to be alive. Um, However, they are incredibly complex. We start with a fairly basic molecular structure, a backbone with those functional groups hanging off of each of the amino acids. They fold together in these three-dimensional shapes, and we can make, again, essentially an infinite number of combinations. And there are different functions for each of those combinations. And we gave you those 9 basic classes, but there are other classes of function, and even within those classes, there are hundreds of thousands of different proteins when we look across all of the diversity of life on Earth. And so we have billions and billions of proteins overall when we talk about all of the species. And some of them are very, very similar, and some of them are quite distinct. And so it's an immense task to answer this question. What is the function of a protein? And even today, mostly this question is answered experimentally. We will observe that protein either in situ, in the cell or in the living organism, or in vitro, right, in the lab, in silico, doing some sort of technology or computer analysis of it. We might even break it down into individual parts and evaluate individual parts. So there are many, many databases that are resources for examining, understanding, and making connections. And I don't expect you to be able to really use this, but it might be interesting and fun for you to look at. So there are a couple of websites. One we'll look at here at the beginning of this discussion. The other one we'll look at the end. So this protein database one is kind of an interesting one. So if you click on the link here, this website will come up. And again, this is I think this is done in the UK, but it's the protein database. It's got kind of a nice user interface. There's information about coronavirus. This is a diagram of the virus with these receptor proteins on the surface. They kind of have a molecule of the month. You can look at three-dimensional structures of different things and data and research on them. So it's a nice database with lots of technical information. So if you want to look up a specific one, you can do that. So we can maybe look up insulin and see if we could find an insulin molecule. All right, so here we go, and we've got research and structural information. You can even come into their little 3D viewer and look at the 3D structure of protein, that protein, all of the different parts nicely labeled. And so it's a nice way to do it. Now, again, much of this understanding, the reason we know this three-dimensional shape, we know the function of this protein, is it's come from observations, from experiments and studies where we're actually looking at the protein itself. We are getting better at being able to take primary protein structure, right, the amino acid sequence, and predict what the function of that protein will be. But it's still a very, very difficult thing to do. And so if given a novel amino acid sequence, we might only have a very vague idea about what the final general job of that protein will be, and maybe even very little idea about the specifics of that job. So we still have a lot of work to do. And most of the time, the way that those predictions are made is by comparing them to other known proteins that have been better characterized. So for proteins, the shape is function. If they are not in their correct shape, they are just simply macromolecules that don't do anything at all. So normal protein has the primary structure that folds into these secondary structures that then fold into the tertiary structure. And for some proteins, then multiple subunits going together to make up the quaternary structure. If we heat this up or have too high or too low of a pH or any one of a number of other environmental factors, we lose this three-dimensional structure. It unfolds so that all we have left is our primary structure. That is called a denatured protein. And a denatured protein doesn't do anything useful for the cells. When you cook an egg, you take all these great energy storage molecules that can be easily used and processed into the chick, and you denature them, and then they stick back together. And that's so why we get this liquid egg turning into a solid hard-boiled egg or fried egg or scrambled egg. And those can be taken into our body and used as nutrition, but to be able to be used, they have to be broken down again to their basic components by our digestive system. So denaturing can occur in any unstable condition, and often for many proteins, there's a very narrow range of this traditional shape. So the reason that fevers can get very dangerous if they get above 105, 106 degrees Fahrenheit is the protein in our cells begin to denature and the cells stop to function, and that can lead very quickly to very severe consequences. So we need to maintain a stable environment and the correct shape. Some proteins have a wider range of conditions where they can work and still maintain their shape, but others are very, very sensitive. Now, we can classify the 20 functional groups and then the conditions, the characteristics that they give to their amino acid. And so we have these different, some overlapping and inclusive. We have a large number of polar amino acids based on their functional group that are here with this large circle. And within that polar one, some of them are small, some of them are a little bit larger, right? So we have polar ones. Do we have a large one? I guess we have tiny, small, and then everything else in between. Some of them have aromatic chains. And again, you don't need to memorize all of these, but just know that we can, to some extent, begin to classify different amino acids based on how similar or different they are from one another. So just as an example, leucine, abbreviated here with the L, and isoleucine are very, very similar to one another. They're both hydrophobic aliphatic amino acids. And so that means that if I was to swap out a leucine for an isoleucine in a protein, it might have a very, very similar impact. In fact, we might have no impact at all if we swipe them out. Valine, which is somewhat related, it is also aliphatic, but it is a little bit smaller, and but it also falls into this hydrophobic category, but it's smaller, is not quite as similar as isoleucine and leucine. And methionine, right, which is the start codon, is similar to them also, but different, even more different from valine. So we could potentially do this. So if I was to substitute, have a mutation, and isoleucine was substituted out for a leucine, that might make very little or maybe even no change. But if I substitute it out something over here, and I don't have all these, remember, I think that's glycine. I don't remember all of my abbreviations. But if I substituted out a glycine for a leucine, that is much more likely to have a more significant impact on that protein because it's a very distinct and molecularly different, functionally different amino acid. So this is an important start if we want to start thinking about models of protein evolution. What happens when I change one amino acid for another? And this is important. I want you to start thinking about this, and we'll do it in some detail later on. We talked about models of nucleotide evolution, which are much simpler because we only have 4 nucleotides, and DNA really only has one job, and that's to code for genes, right? But proteins have many, many different jobs, but they're not completely independent because it's those genes, that DNA, that make proteins. So the evolution of genes can be impacted both by the DNA structure and then also by all these different levels of protein complexity. And so changes at the DNA obviously are going to have an impact to changes in the protein, but not all the time. Sometimes changes at DNA don't impact the protein sequence at all, and so we don't even need to worry about all of this additional level of complexity. So what happens when a mutation results in a different amino acid? We have a change at the amino acid level. Well, again, this is a very complicated question. So as the answer is to many good questions in life and biology also, it depends. So I want you to be able to understand and answer questions about some of the complexity here. And we're not going to be able to do it exhaustively, but we'll give you a big overview here. So number one, which we've already talked about, is what the change is in the amino acid. If we change a isoleucine to a leucine, then the impact might be negligible. If we change an isoleucine into a glycine or another distantly related amino acid, then it might be a big, big change. And it's impossible, well, it's very, very, very hard. We're still trying to figure out to predict ahead of time. If we could predict ahead of time in a complex manner what the change was, we could design proteins from scratch. And we have the technology now, we could design bacteria or yeast if we needed a eukaryotic organism to produce those proteins, and we could make any protein. So for instance, imagine that we could design a protein that would strip hydrogen off of water and release it into the air. and if it could do that using maybe sunlight as an energy source. Now, it turns out there are actually some proteins that can do that, but it's in a very complex molecular mechanism. They can't do it all by themselves. If we could design a protein to do that, we would instantly solve the energy crisis in global warming. We don't really have an energy crisis right now. We have abundant energy, but most of the energy we use is having a negative ecological impact. So we could solve the climate change problem, because hydrogen is a great energy source. When you burn it, the only waste product is pure water, right? And so if we had a ready, easy available hydrogen source, we would solve energy crisis. Maybe some of you should go on and do that. This is not my area of expertise, but I think that this is a huge potential. You could make biogenic hydrogen, right? And there are some organisms that release hydrogen as a waste product, but it's not very efficient, and it's a very, very complex structure. So if we knew ahead of time exactly what a protein could do, we could design them. But it's so complex that the only reason we know is by comparing them to other proteins. And so sometimes people make small tweaks and changes, but we don't really know ahead of time the changes that we need to make to get a desired effect, okay? So it's incredibly complicated, but natural selection and evolution is like this huge laboratory where random changes are made, and then the organism goes forth, lives its life to see if that change is good or bad. If it's good, it's kept. And so all of these dramatic, amazing changes, these incredible wide array of jobs that proteins can do, have been the result of this natural laboratory. Make a random change. It's like tinkering, basically, right? But there's no hand that drives it. It's a random mutate. Well, let's say that there's no intelligent, and I'm going to back up even there because, well, many of you have had my evolution class. And the idea of if God exists is not something that is within the realm of science, right? So at least as far as we can tell, the way that mutation works is that there's no driving force doing it. It's not like a change that says, oh, if I make this change, it'll get better. If I make this change, it'll get better. And so we always see changes that make things better and better and better. That's not the way evolution works, right? You have a random change, and that change might be good, might get that protein working a little bit more efficiently or make it do a slightly different function that's good for that organism. But it's much more likely that that change is bad. But then natural selection works as a filter to filter out all those bad changes. So I digress a little bit here, but random changes through the filter of natural selection result in all of these very, very important, well-adapted proteins that are doing very particular jobs. But we cannot, even with all of our understanding and technology, design proteins from scratch. Now, in addition to the change, another thing that may determine the impact of a mutation is what part of the protein we mutate. If we mutate a part of the protein that's very, very critical for that protein's function, say it's an enzyme with a binding site, mutations to the binding site are often going to have a very dramatic impact on function. Or if I mutate a different part, maybe it's only a very minor structural part of the protein, there might be a lot more flexibility, and we can have lots of changes there that really don't impact the function of this protein, because that's the binding site is the critical, important part of this particular protein. The function and the role of the protein itself might play a role as to the impact of the mutation. So if we have a mutation in lactase, right, which is the enzyme that digests lactose, and so it's not expressed throughout our life, then that might be not really that important of a mutation. We might be lactose intolerant and have to modify our diet at all, but it might only have a very minor impact on us as an organism. If we have a mutation to an eye color gene, again, maybe we get eyes that are a little different shade than any other individual on Earth. And that might have a minor impact on that organism, but it's not going to kill them. And probably, depending on the culture they live in, unless somebody says, oh, look, demon with different color eyes, it's probably going to have only a very minor impact on the way that that human lives their lives. Might be a more major impact for other organisms where eye color is a little more critical. But certainly if we mutate insulin, right, which is critical for correct regulation of our blood sugar levels, then that might be a major, unless it's a part of the insulin gene that's not really important. But if I mutate insulin in a meaningful way, that's going to have a very, very critical impact. So some proteins are much more essential than other proteins. So we need to evaluate that. Now, as we are evaluating all these different aspects, there are other things that we can begin to think about. So I'm going to give you some basic terms, and then we'll, to some extent, explore some of these a little bit more in our final lecture for this unit. So let's just define these and give you maybe a couple of examples. Proteins, particularly enzymes, can either be specific, meaning they have one job and one job only, or they can be promiscuous, meaning that there's more of a range and a little bit of flexibility. They might have off-target jobs. So some proteins are very, very specific, and others have a little bit more leeway and can be a little bit more promiscuous. Robustness versus stability. Robustness means how well a protein is able to do its job despite variability in environment. And some proteins are quite robust and can do their job over a wide range and will continue to do their job, and others are much less and are less stable. So robustness means we can also think about robustness in terms of changes to the amino acid. So in fact, let's back up. More specifically, stability is how well it can do its job despite changes in the environment. Whereas robustness means how much can I change the amino acid sequence and still have it function and do its job. In some proteins, there's a wide range of different amino acids that have mutated in and been impacted by either natural selection or genetic drift, and they still do their job fine. So there's more variability. We might see more allelic variation among these. Whereas molecules that are less robust, there's very little allelic variation because changes lead to impact on the function of that protein. So again, review. Stability, the ability of a molecule to do its job despite environmental variation. And robustness is the ability of a molecule to continue to do its job despite changes to the amino acid sequence. And then we also get these interesting trends in proteins where we can have compensatory mutations. If a mutation occurs that changes the function slightly of an amino acid, natural selection can work to either make that amino acid more common or to remove it from the population. Okay, but sometimes those changes, if they are negative, can still become fixed or common in a population. It's rare, but there are other forces other than natural selection that play a role. So if I have a mutation that fixes a slightly detrimental or slightly negative mutation, so that protein's not doing its job as well as it was before. Now again, normally natural selection would remove those variants from the population, but occasionally because of genetic drift or because of trade-offs in pleiotropic genes, if you don't know that or don't remember that, that's okay for now. But just be aware that there are other things other than just straight natural selection. And so if a negative mutation, a slightly negative mutation is fixed in an amino acid, then later there might be another mutation that can compensate or make up for that detrimental mutation. And so we call that a compensatory mutation. Now we have many, many tools to evaluate proteins and many great modern analytical techniques, but we still rely on some somewhat old school that have been around for many, many decades. One of which is running proteins out onto a gel, electrophoresis of some sort. And so this is actually, it looks like kind of a very busy picture of the night sky, but in reality, this is a two-way gel where we run proteins based on charge in one direction and maybe based on size or some other quality in another direction, and we separate out all the proteins. So every dot here, and the bright dots represent a higher concentration, but every dot here represents a different protein from a biological sample. So for instance, we might take a blood sample and isolate all the proteins and then run them out on a gel and separate them out. So we get an idea of all of the proteins that are present in a sample. So gel just represents one tool that we have to evaluate protein complexity and protein variety in a sample. So for the rest of this lecture, we're going to talk about a characteristic of proteins that we call a domain. And domains can be made up of one or more secondary structures. So domains are really kind of like the sub portions of proteins that allow them to do their job. So let's make an analogy to help you understand what we mean by a domain. If we make the analogy that a protein is like a little molecular machine, think of it like maybe a motorcycle, right? A motorcycle is simply a machine that we use for transportation. There's some proteins that are used for transportation. So that's a pretty good analogy. But within that machine, we have different components that each has a very specific job, right? So you have the wheels and the tires, right? You've got the engine and the carburetor and the signal and the starter motor and all the other parts that make up a motorcycle. Well, proteins are little machines, and so we can break them down into different components that have a specific job. So we may have a portion of a protein that helps to keep it stable in a membrane. Another protein might hang outside of that and serve as a receptor for a signaling molecule. And then we might have a third domain that is inside the cell that catalyzes some molecular reaction. And so in that just kind of very simple example, we have three main domains that would make up all of the parts of a protein. So here's a diagram showing a single small domain, showing a domain that's made up of more than one. And sometimes domains can be made up of parts of proteins that are really far away from each other initially in the primary sequence, but when they fold together in their final tertiary structure, they get right next to each other. So domains don't have to be contiguous in the amino acid sequence, but they do need to be next to each other to perform some function in the final tertiary structure. Okay? And so these can be identified across related proteins. They will fold independently most of the time. Sometimes they need a little bit of help. But given the correct conditions, most domains fold into their correct shape. And they can be observed in the context of, again, these neighboring domains, but we can see different proteins that have sometimes the exact same domain. And so I want you to start thinking about that. If I have protein A with a recognizable domain, and then I find another protein, maybe even in a very, very different part of the genome, right? So maybe the one protein, the gene for it is found on chromosome 1, and then I have a very closely related protein with the same sort of domain in it that's found on chromosome 4. What does that mean? Why are they so similar? We'll come back to that in a little bit. And we can duplicate these coding sequences and kind of mix and match and maybe begin to reconstruct the evolutionary history of these proteins and where they came from. Some proteins only have a single domain. They only do one thing. Those are very simple. Most proteins are much more complex than that. And so they have a combination of domains. So here is just a very small component of a transcription factor that has a DNA binding domain. So all these blue parts are just a small part of this transcription factor. And another part might interact with the cytoskeletal elements to help anchor it, or another part might help it be transported from one place to another. But this part of the protein serves to bind to the DNA, and that's a recognizable domain. And we see this is called the homeodomain. It's a DNA binding domain that can be found across a wide range of different proteins. Now, each domain has its own separate function. And so just like a motorcycle, you could potentially take off one part of that motorcycle, one component, and use it on a different motorcycle, maybe even a different machine, right? So for instance, if I took the headlight off of a motorcycle and put it onto a scooter or car, or maybe even not a vehicle, if I had it in the correct context, it could still serve some sort of a useful function. And so in a way, we can kind of think of domains as these little elements that we could mix and match and potentially have some sort of a useful outcome. So they are little separate functions within the overall function of whatever the enzyme or whatever the protein is. On average, these have 100 to 250 nucleotide range. So if we turn that into amino acids, it's a third, right? Because 3 nucleotides are turned into 1 amino acid through the process of transcription and translation. So on average, we're looking at about 30 to maybe close to 100 amino acids on average. Some are smaller, some are more, to make up a domain. And most proteins, again, have many domains. So we can see, and often in these three-dimensional structures, the domains are color-coded, and we can see the different domains here of, this is a three-dimensional structure of an antibody. Okay? So binding domains are critical for some function for some. We might represent them with molecules. We might have different, this is one single domain, but the different parts of the domain, the secondary structures, are color-coded here to give us the overall complex domain. And as I mentioned, many proteins have very similar domains. So here is a family of proteins that all have some similarities. Okay, so notice that we have this blue domain, and this is looking at just the primary structure without trying to put it in its three-dimensional shape. But in just the primary structure, we can recognize a blue domain. We have these repeated pink domains. We have this light green domain, and so these are all classified as one family because they all have these similar repeated domains. And so we call a protein family with we call a group of proteins with similar domains a protein family. Okay? And it's assumed because of this similarity that they all descended from one common ancestral gene that had that domain. Okay, this is an idea of homology. Hopefully it's a concept you're somewhat familiar with, but it's one we will revisit here in a coming chapter. So ideally I could just say protein families have homologous domains, and you would know what that means. But if not, we'll simplify it a little bit. Protein families are made up of domains that are all descended from a common ancestral protein with that domain. Now, the protein repertoire, which is the number of genes and how different in function those genes are, we find in an organism. So all of the proteins available. The protein repertoire is impacted by three major mechanisms going on at the protein level. One is duplication, and this can be either duplication of a domain. So notice back here I have this pink domain that's duplicated over and over and over again. So that can make a different function. Or we could even have duplication of the entire gene so that we have two copies of that protein. But duplication is the major, in fact, really the only, when we have duplication of the entire protein, it's the only way that we get more and more and more genes. It's very rare for genes to evolve de novo, but they can, and we'll look at that later. We also have divergence of a gene once it's already there. So this doesn't create anything extra. It just makes a new function for a protein. So we might have proteins that diverge in function. So just as an example, insulin in humans and many other mammals has not really diverged in function. So I can take cow insulin and pig insulin, put it into a human, and it works just pretty much exactly the same as human insulin. But if I go too far afield, I might find insulin that is no longer able to function in the same way, and it's diverged. And then finally, recombination. And again, like divergence, recombination doesn't make extra copies or so we have more genes than we had before, but we mix and match different genes via unequal crossing over, or even crossing over. We can mix alleles and end up with a different end protein result. Okay, so recombination also adds to the diversity and types of proteins that we find in organisms. And so here's just another example of a protein family, all with this same PLC domain. And the ancestor maybe only had one, so some of them remain somewhat simple. Some of them have duplicated. Notice these are simplified and a little bit shorter. Some of them have recombined and mixed and matched so that there are domains in between them. So we have this large protein family defined by this domain. Okay? And depending on how divergent and how much these have mutated, it can be sometimes difficult to recognize these. But some of them are so similar that it's easy. We put them all in the same family. So we have the globin family, myoglobin and hemoglobin, that all have this same basic structure. And as we begin to characterize and identify different genes in genomes, we now have hundreds and hundreds of thousands of proteins that have been classified from tens of thousands of species. About half of them we can very easily and readily put into a certain protein family. And by doing that, we've done a little bit of classification, and we can recognize maybe, at least in general terms, what the function of that protein might be. We might need to do much more research to find the exact function, and some families have a little bit of a wider range. They're a little bit more variable in function. Others have very, very kind of narrow specific ranges. But just by identifying these domains, we can put most proteins, or at least a little bit more than half of proteins, into their specific family and maybe get some ideas about their function. And so as we've done this across the different types of organisms, we begin to classify them. So this is looking at different species. Each two-letter abbreviation here is a species. So this is Homo sapiens. This is musculus, and I don't know all of them. I think that one is Drosophila melanogaster. That's Caenorhabis elegans, which is a roundworm. Anyway, we've got a bunch of animals here. We have 2 fungi, I think it is, maybe 3 fungi. All 3 of those, I think, are fungi. and then 2 plants. And what they've done is tried to give us an overview of all the proteins. All of the black part of this bar represents proteins that have family members across the eukaryotic organism. So we might notice that animals have many more. Fungi have a smaller protein repertoire. But we find this domain across the eukaryotic kingdom. The ones in red are ones that are unique to animals. So humans have, I don't know, maybe like 5,000 proteins that are animal proteins. And then the white ones are ones that are for a more narrow group. Maybe this is only for mammals or only for primates or something in white. But notice the vast majority of our genes have similar related groups, even in plants or in fungi. Some of them are animal specific. So the green ones are the ones that are plant specific. And then the fungi have a little fewer, but they have a fewer number that are fungi specific. But in the vast majority of proteins have family members that are very, very widely distributed across the eukaryotic kingdom. And so we can think of a fairly common original repertoire of proteins that has since gone on to diversify and be more complex in some organisms than others, but we still can see these ancient evolutionary connections between proteins. All right, the final thing I want to take a look at here for this discussion is one more database. And we're not going to really use this in detail. It might be kind of fun for you to look at. It's kind of fun just to investigate and play around with. So let's go to that. Actually, I think I already have it open here, but you can go to it if you'd like. But when we are on this website, we can search for different organisms. And I searched for hemoglobin already. One of the fun things about this is you can zoom in and look at the three-dimensional shape. You can highlight different domains. Let's look at one for coronavirus. I don't know if we can search by organism name. Oops. If we can, I probably should at least spell it right? Let's see what happens here. Oh, yeah, we can. So these are different proteins, and I did just a keyword search, so I'm not sure how many of these are actually coronavirus domains. There look like a lot of them. Viruses have rather small protein repertoires, so this is probably not all, and my keyword search maybe has brought up different things. These might be related to immune response to coronavirus or other things, and I don't know it well enough. But we can click on any one of these and then say, oh, here's one in a cow. Here's one in humans. Let's look at that, and we can begin to look at it. So this is a great database. It can show you the amino acid sequence and show you where that domain is in the context of three-dimensional shapes. And so for evolutionary biologists, we might say, hey, let's compare the human one to the cow, see what the changes are. For structural biologists who are interested in how small changes impact the function of a protein, this is a really, really great resource. But it highlights the importance of protein domains and their function as individual components and units of different proteins.